Good evening, everyone. My name is Craig Carter, and today I'm going to be talking about cell gap junctions. So, an overview of cell gap junctions. Gap junctions are clusters of intercellular channels that allow direct diffusion of ions and small molecules between adjacent cells. They are composed of channels of hexamers of medium-sized families of integral proteins, which is connexins and chordates and annexins and prechordates. The structure of gap junctions. So connexin proteins are transmembrane proteins that form gap junctions. They're usually located within the intercellular space around 2 to 4 nanometers thick. Individual connexins assemble intracellularly into hexamers called connexins. This allows it to traffic to the cell surface, which it then docks with connexins in an adjacent cell. From there, it assembles an axial channel spanning two plasma membranes and a narrow extracellular gap. The location and function of gap junctions. Functions have been explored by studying mutations in flies, worms, and humans, and targeted gene disruption in mice. As stated before, gap junctions are located in between adjacent cells connecting the two cytoplasms together. From there, it is able to allow the diffusion of ions, molecules, second messengers, and other cell signaling communication between two adjacent cells. So, diseases. Gap junction has been known to cause skin disease and cardiac diseases. The hereditary skin disorders caused by gap junctions have been caused by gene mutations in the connexins located within gap junctions. This genetic disruption of connexin has also caused cardiac diseases. This has led to a high incidence of lethal cardiac arrhythmias, which has in turn caused sudden cardiac death between humans. So recent discoveries. So one recent discovery of gap junctions is the brain behaviors of rats and mice. So after conducting research, this group found out that mice tighter like gap junctions, and they discovered this because they noticed that mice oscillations were irregular, faster, and more varied in frequency from animal to animal and cell to cell than a rat. And because of that, it provides a new model for analyzing brain activity between related species. It basically showed that even though the rat and the mice are similar rodents, that their neurological structure inside their brain is two totally different systems, even though they're so closely related. So this makes it important for determining the exact frequency of the oscillation in the brain. So my question for today is, how do gap junctions react to passive molecules that don't normally travel between its two adjacent cells?